Hello, we start discuss the immune system organ and tissue. So lymphoid tissue. Immune system, like any other system in body, includes organs, tissue, cells, molecules, and sometimes fluids. The tissue of the immune system are called lymphoid tissue. The cells of the immune system are all blood cells except the red blood cells, erythrocytes. The molecules of the immune system are antibodies, complement, cytokine, chemokines, etc. The fluid of the immune system is called lymph. The lymph is plasma component. Overview of the immune system. Organites is similar to nervous system. Cells of the immune system, immune cells found throughout the body, but also found in special organs. Cells, lymphocytes, macrophages, monocytes, dendritic cells, Langerhans cells, granulocytes, all arise from pluripotent hematopoietic progenitor cells in bone marrow. Organs, lymph nodes found in various locations, thymus, spleen, these constitute the lymphoid organs. Immune system organs and tissue. This picture describes us about all peripheral lymph nodules. We have left lymph ducts, thymus as a central organ, bone marrow, appendix, Peyer's patches, lumbar nodules, iliac nodules, inguinal nodules here, spleen here, axillary nodules, intercostal nodules, thoracic duct, cervical nodules, higher subclavian vein, etc. Lymphoid tissues. Other sites were there: generation, maturation, habitation, and activation of the immune cells take place. Lymphocytes. Major subtypes are T and B cells, responsible for immunological memory. T cells mature in thymus. B cells a vein bursa of Fabricius but mammalia fetal liver and bone marrow. Cells participate in cell medular immunity, regulation response. B cells synthesize antibodies. Natural killer are macrophages similar to T and B cells, are cytotoxic in absence of prior stimulation. This picture display about granulocytes and have between erythrocytes. This maybe look like as lymphocytes or monocytes, lymphoid cells. T and B cells have specific antigen receptor which play roles in developing immunological memory and in specificity of the immune response to antigen. Both T and B cells secrete proteins called cytokines, which form the communication system among the between cells and cells type. T versus B cells. Antigen receptor and receptor immunoglobulins. T cells related to immunoglobulin. B cells is membrane bound. Antigen recognition in context of MHC major histocompatibility complex on antigen presentation or accessory cells, B cells can recognize antigen alone. Functional subsite: T helper and T cytotoxicity cells, cytolytic. B subset of B cells not different in function. Secrete: cytokines, immunoglobulins, and cytokines. Surface markers CD4 and CD8 respectively. For T helper have CD4, T cytotoxicity have CD8, among many others. Immunoglobulins have different subtype. When antigen then activated, become proliferation, become lymphoblasts. Activated lymphoblasts become plasma cells. Co-stimulation required, T cells yes, B cells no. Two major types of T cells: accessory molecules, CD4 and CD8 T helper. T helper split two types: T helper 1 type, effector cells, and work as cellular immunity, and T helper 2 type activate B cells and production a specific antibody, immunoglobulins, combined with class 2 and class 1 MHC major histocompatibility couple, respectively. Class 2 bind and work with CD4, class 1 work with CD8. Function: cytokine production help for B cells and cytotoxic T cells, and CD8 cytokine production, cytotoxic lysis of infected or tumor cells. Monocytes and macrophages. Monocytes are immediate macrophages circulated in blood, accumulate at sites of inflammation. Monocyte macrophages may differentiate in tissue in absence of antigen. Kupffer cells in liver, osteoclast in bone marrow, on bone, glial cells in the nervous system, or differentiation in response to antigen. They are antigen presenting cells and cooperate with B and T cells in mounting immune response. Also, phagocytes, microbes, contain bactericidal mechanism. Monos and macrophages continue. Express a myeloid receptor CD14, which serves as recognition molecules for wide variety of bacterial enveloped molecules, such as lipopolysaccharide gram-negative organism and component of mycobacterium and gram-positive cell walls. Ligation of this receptor leads to macrophages activated. Although they are activated by T cells derived cytokines, leading to increased phagocytosis and microbial activity, increased activity of degradation enzyme, nitrogen and oxygen free radical production, and prostaglandins, etc. Note: T cells derived cytokines increase the antigen presenting activity of macrophage, which in turn are able to present antigen to T cells. This cycle will continue as a positive feedback loop until the antigen is eliminated. Dendritic cells. Dendritic cells are antigen-presenting cells for primary first-time adaptive immune response. also continued major component of the innate immunity system and the bridge to adaptive immunity. Two sites of origin: plasmacytoid dendritic cells, periphery, spleen, and myeloid dendritic cells, bone marrow. So far, no major function difference noted. The dendritic cells, that is not dendritic cells. Follicular dendritic cells are found in primary, secondary lymphoid follicles, but aren't of the same organ as plasmacytoid or myeloid dendritic cells. In fact, the exact origin is unknown, though they may be myeloid, play a role in the controlling B cells response, this areas FGS. Granulocytes, polymorphonuclear leukocytes or neutrophils. Predominant type of white blood cells, rapidly migrate to sites of infection or inflammation. Phagocytic, they have specific enzyme pathway for enhanced bactericidal action and also called azurophilus granules due to blue-stained azurophil granules. This picture describes the polymorphonuclear lymphocytes, neutrophils, and monocytes. For example, this was different looking up to microscope. Basophilic and mast cells have basophilic granules which contain mediators, especially of allergic response, histamine, heparin, etc. Basophilic circulate mast cells found in tissue. It's different between them. Eosinophils have granules that stain red with eosin, mediate late phase of allergic response, active in immune response to parasites, tumor, antibody-dependent cells mediate cytotoxicity. Granules contain toxic proteins of higher pH. Organs of the immune system, primary lymphoid organs. Primary lymphoid organs are where lymphocytes arise and mature in the absence of antigen stimuli. They are the bone marrow and thymus. Bone marrow, source of all hematopoietic progenitor stem cells, site of B cells maturated post-birth in mammals, hematopoietic stem cells, and stromal stem cells in bone marrow. Schematically displayed lymphoid system, peripheral lymphoid system, and these two primary lymphoid organ, thymus, and bone marrow, and peripheral organ. Primary lymphoid tissue, the sites where the blood cells generation, hematopoiesis, and maturation take place. Hematopoiesis take place in yolk sac, first 5 weeks of fetus age, in fetal liver, 5-8 weeks of age, in the whole bone after 4 months of fetus age. In adults, the hematopoiesis take place in the flat bones only, sternum, scapula, skull, pelvis, bone marrow, the site where all blood cells generated and maturated except T cells maturation take place. So display primary and secondary two lymphoid tissue, Walder's ring, and this about schematically to jog back fetus, early stage, middle stage, and later, and bone marrow to adult. Primary lymphoid organs thymus. The thymus is a site where lymphoid cells undergo maturation and education into T cells prior to release into the circulation. This process allows T cells to develop the important attribute known as self-tolerance. The thymus is found in the thorax, in the anterior mediastinum. It gradually enlarges during childhood, but after puberty it undergoes as a process of involution, resulting in the reduction in the functional mass of the gland. It continues to function throughout life, however. The thymus is arranged into the outer cellular cortex, mature, and move to the medulla, from where mature T lymphocytes enter the circulation. Young thymus look schematically like that. Hassel's thymus corpuscle, founded layer, packing lymphocytes, thymocytes, medulla light, cortex dark, and connective tissue small concentration. Thymus young to old lymphocyte traffic. Vessels from bone marrow as pre-T cells, cortex for positive selection, medulla for negative selection, those surviving T cells to live via vessels, to sit to node, spleen on mucous membrane. Acquired immune cells trafficking, lymphocytes traffic, from bone marrow to thymus as pre-T cells, from cortex to medulla in thymus, from thymus to cytonode, spleen or mucosa, from lamina propria into epithelium, from follicles to elsewhere in mucosa, from lymph to blood, from blood to lymph node, recirculation as a high endothelial win. Dendritic antigen presentation cells from tissue to lymph to node. Thymus old look like that, more present as a connective tissue, less cortex, less medulla, more Hassel's thymus corpuscle, and equal involution, this structure. Young thymus like that. Dendritic cells, myeloid, Langerhans cells, lymphoid, plasmacytoid, dendritic cell, monocyte-derived dendritic cell, different. They have CD or cluster differentiation receptor similar as 11 subtyped only C, A, B, C. Look like as similar as all location for this CD11. Lymphoid progenitor, natural killer, bone marrow, pre-B cells, B cells, production thymocytes, and production T cells. Schematically, myeloid progenitor through erythropoiesis forming erythrocytes, through thrombopoietin production platelet cells, melanocyte progenitor through growth factor, a lot of growth factor, and mast cells eosinophils production through colony-stimulated factor, interleukin 3, and interleukin 5 activated and production eosinophils. Interleukin 5 produced self-eosinophil and co-stimulated themselves. Monocyte progenitor scheme. This stem cells differentiation have CD, different CD, and antigen presentation in lymph nodules look like that. Pair patch present in gut, a lot of component lymphoid tissue. Secondary lymphoid organs. There are the peripheral lymphoid organs, lymph nodules, spleen, tonsils, adenoids, and lymphoid tissue associated with other organ systems. Multiform mucosa associated lymphoid tissue, GALT for gut associated lymphoid tissue, BALT for bronchus associated lymphoid tissue, and SALT in skin associated lymphoid tissue. Lymph nodules filter lymphatic fluids, site of antigen presentation, and cell traffic. Lymph nodules have a fibrosis, a capsule from which tubercle extend towards the center, forming a framework for the lymphatic sinuses, blood vessels. And parenchyma, cortex, paracortex, and medulla sinus. Lymph nodes continued. Function of the structural elements of lymph nodules. The lymphatic system is a series of vessels which drain and filter the tissue fluids. Lymph enters the node via efferent lymphatic path. This one passes through the sinuses linked with macrophage and leaves via effector lymphatic wings. Effector lymphatic. Ultimately, all drain into the portal vein. Lymphocytes enter the node primarily from the blood via higher endothelial veins and leave via efferent lymphatics. Dendritic cells migration from tissue enter the node into the T cells areas. B cells entering nodes from blood must cross the T-rich area in transit to the B cells rich area. Thus optimizing T-B cooperation. The B cells rich area contain mature, resting B cells organized into structures around follicles, dendritic cells, primary follicles. Lymph node parenchyma is made up for three components: cortex, paracortex, medulla. Cortex B cell area. B cell enters the lymph nodes via a HEAV, higher endothelial veins, and pass into the follicles. If activated by antigen stimulation, they proliferate and remain in the node. Unstimulated B cells, however, pass out rapidly from the node to return to general circulation. Activated B cells within the lymphoid follicles are known as follicular central cells. The pale stained central area of the secondary follicles is known as the germinal center. In this is surrounded by the mantle zone consisting of small naive B cells and few T cells. The follicle central cells within the germinal centers consist of cells with cleaved nuclei, centrocytes, and cells with large, more open nuclei and several nucleoli, centroblasts. Stimulated mature B cells responding to antigen change into centrocytes and then centroblasts. The centroblast leaves the follicle and pass to the paracortex and medulla sinuses, where they become immunoblasts. The immunoblasts divide to give rise to plasma cells or memory B cells, which are ready to their next encounter with specific antigen. B cells alone aren't able to mount immune response. They are assisted by accessory cells, sinus macrophages, highly phagocytes, tangible body macrophage ingest cellular debris in germinal centers. Marginal zone macrophage found beneath the subcapsular sinus, follicular dendritic cells. Paracortex T cells area. The paracortex contains lymphocytes and accessory cells along with supporting cells. It's the predominant site for T cells within the lymph node. The various types of T cells enter the node from the blood via the higher endothelial veins. When activated, they form lymphoblasts, which divide to produce a clone of T cells responding to a specific antigen. Activated T cells then pass into the circulation to reach peripheral sites. Accessory cells, interdigitated cells, are numerous in the paracortex and act as antigen presentation cells. Medulla. The medulla is comprised of large blood vessels, medulla cords, medulla sinuses. The medulla cords are rich in plasma cells, which produce antibody that pass out of the node via the efferent lymphatic. Macrophages are also numerous within the medulla. Lymph passes into the node through the efferent lymphatic into the marginal sinus, though the cortical sinuses to reach the medulla sinuses before leaving via the efferent lymphatic. Particular matter in the lymph is removed by macrophages. Antigens are taken up by antigen-presenting cells, and these facilitate the specific immune response. Less than 10% of lymphocytes enter the node in the lymph, the large majority entering from the blood via the higher endothelial veins. There are two distinct components of the spleen, the red pulp and white pulp. The red pulp consists of a large number of sinuses and sinusoids filled with blood and as responsible for the filtration function of the spleen. The white pulp consists of aggregates of lymphoid tissue and is responsible for the immunological function of the spleen. The spleen serves two major functions. It's responsible for the destruction of old red blood cells It's a major site for mounting the immune response. The spleen behaves similar to lymph nodes, but instead of filtering lymph, it filtered blood. Blood entering the spleen travels through progressively smaller arteriole until it's deposited in the red pulp, where the red blood cells are processed. The interface between periarterial lymphoid sheaths and blood is a region of intense phagocyte activity and sets the stage for immune response. The immune reactivity of the spleen is especially effective for dealing with blood-borne antigens such as bacteria. Inside the spleen, red pulp. The red pulp has a complex system of blood vessels. Within it arranged to facilitate removal of old or damaged red blood cells from the circulation. A small proportion of splenic blood flow passes through more rapidly without undergoing filtration. Inside the spleen continued white pulp. The white pulp contains T cells, B cells, and accessory cells. There are many similarities with lymph node structure. The purpose of the white pulp is to mount an immunological response to antigen within the blood. The white pulp is present in the form of a periarterial lymphoid sheath. This sheath contains B cells, follicles, and T cells. At the edge of the T-zone is a region known as marginal zone, where large lymphocytes and antigen-presenting dendritic cells are located. Gut-associated lymphoid tissue, or GALT. This is comprised of tonsils, adenoid, Walder's-Rhine, Pear patch, lymphoid aggregates in the appendix and large intestine, lymphoid tissue accumulation with age in the stomach, small lymphoid aggregate in the esophagus, diffusely distribution lymphoid cells and plasma cells in the lamina propria of the gut. Large aggregates of GALT have distinct B cells, follicles, and T cells areas. Antigen-presenting accessory cells are also present. Pear patches are larger aggregated of lymphoid tissue found in the small intestine. Lymphocytes form domed follicles of B cells surrounded by T cells. Some epithelial cells have complex microfolds and in the surface, known as M multi-fenestrated arrangement cells. They collect antigen. Pear patches facilitate the generation of the immune response within the mucosa. B cells, precursor and memory cells are stimulated by antigen in pear patches. Cells pass to the mesenteric lymph nodules, where the immune response is amplified. Activated lymphocytes pass into the bloodstream via the thoracic duct. These cells then home in the gut and carry out their final effector function. Here or higher endothelial veins aren't present in pear patches. and the mechanism by which cells home in on mucosal sites is unknown. Cell surface molecule known as aderesin may have a role. This schematically about structure, pair patch and TNB zone, follicles, dendritic cells, centroblast B cells, lamina propria, villi, M cells, very important to immunological function, follicle-associated epithelium, nuvoc lymphatic, and higher endothelial wings. Mucosa-associated lymphoid tissue, MALT, mucosa. In addition to the lymphoid tissue concentrated with the lymph nodules and spleen, lymphoid tissue is also found at other sites, most notably the gastrointestinal tract, respiratory tract, and urogenital tract. MALT consists of aggregation of lymphocytes, macrophage, dendrit cells, and other accessory cells. In the gut, these aggregates are scattered throughout the lamina propria, although pairs patches, which resemble lymph nodules in that they have geminal, central, and B cells-rich follicles are also present in the gut. Skin-associated lymphoid tissue. Skin is an active participant in host defense. It has a capability to generate and support local immune response, inflammatory response to foreign antigen that enter the body via the skin. Cells of SALT include keratinocytes, Langerhans cells, immature dendritic cells found in skin, intraepithelial T cells, and melanocytes. Langerhans cells form a continuous epithermal mass walk. They capture antigen, then migrate to draining lymph nodules, where they act as antigen presentation cells. The majority of T cells are found in the dermal layer of skin. This picture about skin and distribution of the cells, epithelium and derm. This Langerhans cells, derms at this interesting dermal dendritic cells, and a lot of monocytes. Antigen presented in skin infected in immature dendritic cells and mature dendritic cells. Low surface MHC2 type and high surface MHC2 type, higher co-stimulation, present as low co-stimulation as a CD8T. Structure. Skin as epidermis, dermis, hypoderma, and mast cells, dendritic cells, endothelial cells, keratinocyte, Langerhans cells. Animal skin separates the inner wall of the body from the largely hostile outside wall and is actively involved in the defense against microbes. However, the skin is no perfect defense barrier, and many microorganisms have managed to live on or within the skin as harmless passenger or as disease-causing pathogens. Microbes have evolved numerous strategies that allow them to gain access to layers underneath as epidermis, where they either multiply within the derms or move to distinguish destination within the body for replication. Number of viruses, bacteria, and parasites use arthropods vectors like ticks or mosquitoes deliver them into the domes, while taking their blood meal. Within the domes, successful pathogens subvert the function of the variety of skin resident cells or cells of the innate immune system that rush to the site of infection. Inflammation induces dendritic cells and matured dendritic cells. Electron microscopy 4 hours initiate activating before 10 hours and 22 hours activated dendritic cells. As it's immature dendritic cells and mature dendritic cells look like. Lymphoid system and primary and secondary T thymus from thymus T cells and peripheral organs differentiation to T helper and T cytolytic cells. Bone marrow B cells in germinal center have B activated and change to plasma cells and produce antibody. Antigen presentation cells, a lot of macrophage dendritic cells and dendritic cells and secondary and natural killer. Lymphocyte recirculation. Lymphocytes and some monocytes can recirculation between lymphoid and non-lymphoid tissue. This helps lymphocytes to be exposed to the antigen which they recognized and is valuable for distribution of effector cells to sites where they are needed. The recirculation is a complex process dependent on interaction between the cells of the immune response and other cell types such as endothelial cells. Naive lymphocytes move from the primary to secondary lymphoid tissue via blood. Activated lymphocytes move from the spleen, lymph nodules, and other lymphoid tissue into the blood and then to other lymphoid and non-lymphoid tissue. Antigen presentation cell may carry antigen back to lymphoid tissue from the periphery. The complex pattern for recirculation depends on the state of activation of lymphocytes, the adhesion molecules expressed by endothelial cells, and the presence of chemotactic molecules which selectively attract a particular population of lymphocytes or macrophages. Induced dendritic cells activated dendritic cells go to lymph nodules and activate this as an antigen presentation site. This picture, B cells follicles staining and T cells brown CD3 staining. The antigen receptor molecules, there are 3 groups of molecules. This specifically recognize foreign antigen for the adaptive immune system, B cell receptor or antibody T cell receptor major histocompatibility complex. This cluster of genes is known as a human leukocyte antigen. MHC major histocompatibility complex, this group of antigen receptor is represented by the protein encoded by the MHC genes located on the chromosome 6, and it's known as a human leukocyte antigen. There are 2 main classes of molecules which were initially named because of their role in tissue histograft rejection compatibility. Class 1 MHC molecules are found essentially on all cells except red blood cells, erythrocyte. And class 2 major compatibility complex molecules are found chiefly on antigen presentation cell as macrophage, monocytes, B cells, dendritic cells. The main function of MHC molecules is to present antigen T cells. We continue on later immunological topic. See you.